Hi, Ben. Thank you so much. It's my pleasure to talk a little bit about nutrient assimilation and homeostasis from the perspective of war and peas. Peas is a play on peas. I won't be talking about peas, but I'll be talking about plants. So I thought that was a nice title. And so nutrient assimilation in plants, who cares about it? Why is this important? I don't think a lot of people realize that plants serve as the main mechanism or the conduit through which nutrients pass from an inert geophysical state, from the soil, into living organisms, into the biosphere, to the main conduit for that process. And I like to show this cartoon to my plant phys students because it really kind of helps you understand when you look at a plant, there's a lot that's going on that's really important in terms of how the plant interacts with the environment and how the plant interacts, serves as a nutrient source for us. And so this is important because what you eat is a direct reflection of what sort of nutrients are available to the plant. So when you think of nutrients, essential nutrients, these are molecules that are intrinsic components or of the structure or of the metabolism of a plant. And when there's a deficiency in those nutrients, it can lead to severe abnormalities in growth, productivity, production, and just the overall decrease in our health because we're not eating nutritious plants. And so when you think of the essential components of the plants, you see many of them are listed here. Many of the nutrients that we think of are actually derived from inorganic ions from the soil. So nitrogen, calcium, iron, I'll talk a little bit about. And hydrogen, oxygen, and carbon are actually derived from water and from oxygen. And so they're not considered actually minerals that are acquired from the soil. So I just realized, I think I got this from online. I think there's actually a mistake in how this is labeled. So I think this is non-mineral and mineral. So I think they made a typo there in where I got this from online. But suffice it to say, hydrogen, oxygen, and carbon are derived from the air, whereas minerals are derived from the soil itself. So we think of things like iron and nitrogen. And so this is really important because the minerals that we acquire, for example, nitrogen is the fourth most abundant mineral or substance in plants. Right? And so nitrogen is really important because it's composed, all of the amino acids, which are part of the proteins that make up the plant, contain nitrogen. They contain hydrogen and oxygen and carbon, but as I said before, those are considered, those are not derived from the soil. So when we think of nutrients that are derived from the soil, the most important one is actually nitrogen. And the interesting thing about nitrogen assimilation is that it requires a lot of energy. And the reduction of nitrate to forms that are actually in amino acids, it can acquire or utilize 12 molecules of ATP per amino acid. That's a lot of energy to go from, nitrogen, molecular nitrogen in the atmosphere to nitrogen that's actually fixed into amino acids. It's a lot of energy. And so much energy is required in this process that if you run those reactions in reverse, they're actually explosive. And it is for that reason that nitrogen and actually many explosives are based upon the rapid oxidation of nitrogen as well as sulfur compounds. So for example, TNT is actually highly contain lots of nitrogen, and the oxidation of the nitrogen in this compound is what leads to this high release of energy. So this just tells you how much energy is required to actually fix nitrogen into a source like amino acids. So as I said before, nitrogen is the fourth most abundant element in plants. Right? It is a component of nucleotides, amino acids, proteins, many things that you can think of actually require nitrogen. And then one of the interesting things about nitrogen is that most of the atmosphere is actually molecular nitrogen. Right? It actually forms a stable triple covalent bond between another nitrogen molecule. And this bond is very, very strong. And it takes a lot of energy to break that bond. And it takes a lot of energy to break that bond. That's required before you can actually, quote unquote, fix that nitrogen into basically amino acids. And so it's the fourth most abundant element in plants, and it's one of the most abundant components of our actual atmosphere. Right? So our atmosphere is composed of 70% nitrogen, 21% oxygen, and carbon dioxide. And so how does this nitrogen go from, molecular nitrogen is floating around in the air to actually being a part of our plants? And as a consequence of their involving being a part of plants into our bodies, and which are then utilized to make our proteins. And so, as I said before, it takes a lot of energy to break that bond. And so the majority of nitrogen is actually fixed through bacteria. And so there are nitrogen fixers that are free living, and then there are nitrogen fixers that form symbiotic interactions with plants. And so the enzyme that's and so then once that nitrogen is actually converted into a form through these free living or symbiotic nitrogen fixers, it's converted to ammonium, and then that ammonium is converted to nitrite, and then both ammonium and nitrate can then be transferred and then uptaken directly by plants. Mm-hmm. And so the enzyme that actually converts the molecular nitrogen to ammonium is actually a nitrogenase complex that's found in bacteria that fix the nitrogen. And so here is kind of give you an idea of what it actually does. And here's one of the enzyme structures. And so these free living nitrogen fixers, just kind of a cartoon, are found in soil. And as I said before, they form symbiotic reactions with plants like peas, as well as probably most commonly known nitrogen fixers are those that form nodules with soybeans. And so then once that nitrogen is converted to ammonium and then converted to nitrate, that nitrate is then transferred into plant cells and then converted to nitrite and then eventually into ammonium. And then that ammonium is then inserted basically into molecules to make a variety of amino acids shown here. So this is a lot of work that the plant has to do to convert nitrate or ammonium into amino acids. And as I said before, there are specific transporters for nitrate as well as specific transporters for ammonium. But there's a lot of nitrogen that can actually, nitrogen can also be fixed through industrial mechanisms. And so this is kind of one of the ways that the idea of war comes into play. So during World War I, the Germans had lost, were losing because they did not have access to munitions. And the Allied forces had access to Chilean saltpeter mines, shown here. And so the Chilean saltpeter mines were actually for a source of nitrates, and these nitrates could then be used to make explosives and, like I said, munitions. And so the Germans were losing, and they wanted to develop a mechanism to fix nitrogen using high heat, pressure, and metals. A guy named Dr. Fritz Haber in 1918, early 1900s, figured out a way to combine these things to form ammonia, which can then be oxidized to make nitrates and nitrites for explosives. And so using this mechanism, he worked with Karl Bosch to industrialize it. And then they were able to basically prolong World War II using this industrialized form of nitrogen fixation. Not only was this form of nitrogen utilized to make munitions, it was also a great source for as a plant fertilizer. Mm-hmm. And so these fertilizers that were being produced using this industrial mechanism actually contributes or correlates with a major increase in the global population. So much so that a good chunk of the nitrogen that's a part of your body is actually derived from the Haber-Bosch process, because the nitrogen that fertilizes a lot of our plants today are actually derived from that process, from industrial nitrogen fixation. And so I like to talk about this with my class, because it's a really good example of, of showing how different pieces of science can come together to really impact your life. And this is all due to basically an effort to win a war. I also kind of like to point out here that another reason why nutrient assimilation is important is because the herbicide industry relies on creating ways to affect plants that are really specific to plants. And this image shows you a lot of the metabolic processes that occur to convert sugar molecules during metabolism. And so as you go through the citric acid cycle, you're actually creating byproducts that are used to make amino acids in plants. And some of these pathways are specific to plants and can only be produced in plants. So this is one of the reasons why plants are so important to our diet, because we can produce a lot of these amino acids. And so many herbicides actually target some of these plant-specific pathways. So for example, Roundup targets this branch of the pathway that creates amino acids. And another important pesticide, Liberty, affects another branch of amino acid synthesis. So I also like to kind of point this out. Another reason why nutrient homeostasis is important in plants, because understanding that enables us to create really effective herbicides. And so I've spent some time talking about nitrogen fixation and why that's important. But my work really kind of focuses on iron homeostasis, which is an important micronutrient. And micronutrients are actually required in much smaller amounts than the macronutrients, such as nitrogen, phosphorus, and potassium. And it is, again, one of those elements. It is a mineral that's actually required from the soil. Mm-hmm. And so, unlike the assimilation of nitrogen, which requires lots of energy on behalf of the plant, the assimilation of iron involves the formation of, it seems rather much simpler, which is the formation of complexes with organic compounds, for example. So you have, for example, a porphyrin ring, which again ironically contains nitrogen. And then an enzyme will basically pop iron into this ring. And then this ring actually becomes a source of iron for many other proteins. So iron can be assimilated into heme-containing proteins. So this is an example of heme ring. It could also go into iron sulfur clusters, and these heme-containing proteins and iron sulfur proteins are involved in many processes. So, for example, these protein complexes, many of them actually contain iron as either in an iron sulfur or in heme form. And these proteins, this is actually showing you some of the protein complexes involved in photosynthesis, right? And so what you see is a lipid bilayer that's found within the chloroplast. And these protein complexes will, one of which, photosystem 2, actually facilitates the absorption of light, the conversion of resonance energy to cause the movement of electrons down an electron transport chain that consists of cytochrome B6F, plasticinin, and another protein complex called photosystem 1. The movement of electrons down this chain then builds up a proton motor force that's utilized by ATPase to make ATP. This process also leads to the synthesis of NADPH. This ATP and NADPH are then utilized by the Calvin cycle to convert CO2 and oxygen into complex carbon molecules, like eventually sucrose and glucose. And so this process takes place within the chloroplast. Image is shown here. And these chloroplasts are organelles that are found within plant cells, predominantly in the green parts of the plant. And I like showing this because it just shows you how that there are the thylakoid membranes, which are these membrane systems that are found within the chloroplast, and embedded within these thylakoid membranes are these enzymes that I've indicated here. And these enzymes are not only involved in helping to make ATP and NADPH, they're also involved in converting oxygen, in evolving oxygen from water. So this is the source of oxygen on Earth. Okay? And this is the source of glucose, sucrose, and the complex carbon molecules that enable the plant to grow. And then we consume these sugars, right, as well as the oxygen that the plants produce, and then we utilize that to make our own ATP in the form of respiration. And this ATP is the energy currency that allow us to survive. And so iron is not only an important component of the linear electron transport chain involved in photosynthesis, it's also an important part of the proteins that make up the respiratory electron transport chain in our mitochondria. Right? So they're actually important for facilitating the synthesis of ATP in the mitochondria. So this is an image of mitochondria. And this is just showing you one of the reasons why iron is so important, because without iron, sufficient amounts of iron, this process doesn't take place. And as I'll show you, it doesn't take place effectively with decreased amounts of iron availability. And as I'll show you in a few slides, this impacts the health of many people across the world. So iron is assimilated into heme-containing and iron sulfur-containing and other iron-containing proteins involved in human respiration and many, many other processes. So iron is important for both plants and animals. And so iron deficiency in humans is a global problem. Approximately 30% of the global population suffers from iron deficiency. And this is particularly prevalent in developing, what's considered developing countries. And it affects women who are of childbearing age, children, and the elderly, particularly. And it's been thought to lead to an overall 10% decrease in overall human productivity, because it is the most prevalent nutritional disorder in the world. So plants are the most common source of iron. Most people, meat is considered the best source of iron, but a lot of people don't eat meat for socioeconomic reasons and for financial reasons. Meat, some people just don't consume meat, and so iron is acquired from plants from a lot of people. And so we see a lot of the iron-rich foods here, and I'd like to point out again, I'm back to peas. And so we study iron homeostasis in the plants because we really wanted to try to dig into a little bit, understanding how plants respond to iron deficiency. Can we learn about the genetic mechanisms that plants utilize to respond to low iron availability? And can we manipulate it so that we can create plants that A, can tolerate iron deficiency better, and B, can actually acquire more iron, and that iron is actually biologically available for human consumption. And so iron deficiency, like I said before, in humans, it leads to just overall decreased growth, decreased immune response. In plants, it leads to, that's kind of sort of the same thing. The plants become chlorotic because they can't make enough chlorophyll. Iron is required for chlorophyll synthesis. So you see this intravenous chlorosis. And in the plant that I work on, called Arabidopsis thaliana, which is the model plant species, it's like the Drosophila or the mouse of the plant world, iron deficiency leads to changes in root morphology and changes in shoot morphology and chlorosis and eventually death. So what's up with iron? It's one of the most plentiful elements in the Earth's crust. It's the fourth most abundant element in the Earth's crust. The problem is that it forms these insoluble ferric oxides that are only available through the weathering of rock. And in calcareous soils, it's even more poorly available. And so plants have figured out ways to solubilize and increase iron uptake. And there are two different ways that plants do this. And so if a plant can't do that, for example, we have two varieties of soybean that one variety is called iron deficiency chlorosis susceptible, and one variety is iron deficiency chlorosis tolerant. And this variety is unable to mount the strategy one response that people have named it, that enable plants to solubilize and increase iron uptake. And again, so this picture is a little bit redundant with another picture. And so I study it. So this is what iron deficiency chlorosis looks like in soybean, and I study it in Arabidopsis thaliana. And we grow them quite often on these Petri plates that allow us to do really nice molecular work. And so what is the strategy one response? So I'm showing you this image. This is from a review that we wrote last year. I wrote with Dr. John McDowell at Virginia Tech last year, who works on pathogen response. And we were interested in pathogen and how pathogens might impact iron homeostasis and vice versa. But I want you to focus on the left side of the slide for now. So here's an Arabidopsis thaliana seedling And in response to iron deficiency, Arabidopsis and many other dicots will mount what's called the strategy 1 response, as I mentioned before. And so what they do is they pump, so if you focus in on the roots, in the epidermal cells, the outer layer of the roots, they'll actually pump protons into the rhizosphere, which is the region right outside of the root. All right, this changes the pH and makes iron more soluble. And then this more solubilized iron is then reduced because I said before it forms ferric oxides. These ferric oxides are reduced from the ferric form to the ferrous form, and then the ferrous form of iron can then be transported from the rhizosphere into the epidermal cells, where it then travels into the vasculature, and then from the vasculature in the root, it can then travel into the shoot, and then it can be released from the xylem, which is basically the parts of the vasculature of the plant in which water and other nutrients can move from the root into the shoot. And then they're released into the leaves, the cells, the mesoville cells of the leaves. And then inside of those cells, they can be incorporated into, for example, into chloroplast and mitochondria and stored in the vacuoles for the metabolic processes that I described earlier. And so this kind of gives you a brief snapshot into how iron is incorporated into plants, assimilated into plants. And this picture, another kind of a brief shout out to war or battle, shows you that plants are often competing for iron with other plants, but they're also competing with iron from bacteria, if there's a bacterial infection, or fungi, if there is an infection from fungi. There are beneficial bacteria, and there's a lot of research that's been going on now to try to figure out how beneficial bacteria facilitate iron availability and uptake in plants. But then there are the harmful bacteria and pathogens, and they actually can impact the plant in a variety of different ways that I won't go into in this talk. If you're interested, you can take a look at the many reviews that have been written recently that kind of looks at how different types of pathogens will induce different types of cell death to facilitate their own livelihood. And some of these responses actually are very similar to a low iron deficiency response. So you can look into that if you're interested. And so again, that's another a shout out to the idea of battle or a war. And so I was particularly when I was a postdoc. I was a postdoc in Philip Benfey's lab, and he was really interested in cell sorting and trying to look at regulatory networks that were involved in individual cell types. And so I really became intrigued at the idea of using cell sorting to look at what was happening in individual cell types of the roots with respect to iron deficiency. And when I first started working in the field, we knew that one of the master regulators of the iron deficiency response was FIT. And we knew that FIT was a master regulator that controlled the 3 main steps of the strategy 1 response that I mentioned earlier, the proton extrusion, the iron reduction, the transport. These 3 genes were controlled by this master BHLH protein called FIT, which formed binding partners with other homologous proteins. So I became interested in trying to figure out what are some of the other regulatory mechanisms involved in this process. So and I'm dating myself here. This was many years ago. So we did a bunch of microarray analysis to try to figure out what sort of genes were differentially expressed at different time courses, in specific developmental zones of the root, and in specific cell types. And then we queried some of the genes that showed the most differential expression, and 2 of them have become kind of the foundation for the work that we do in my lab today. And I have this little arrow kind of pointing to them to kind of segue to the next slide. One of these genes we named Popeye. It shows decreased, and so these plants are grown on media that has an iron chelator in them, so they're effectively exposed to iron deficient media. And so these plants are suffering from iron deficiency. And Popeye mutants, loss of function mutants, exhibit decreased tolerance to iron deficiency. And Brutus exhibits increased tolerance to iron deficiency. And so when we saw these 2 phenotypes, we were like, oh, what should we name them? And I like the idea of naming them Popeye and Brutus because, and I'm again dating myself here, I'm Generation X. I grew up on Popeye cartoons, and we like the idea of naming it Popeye because Popeye liked spinach and iron. I hate this is not a live audience because usually I get a chuckle at this point, so I just assume people are chuckling. I don't know. And so we named it Popeye and Brutus, and so the synonyms are PYE and BTS. And I gave this talk at a high school, a version of this talk at a high school, and I had a student come up to me, and she was like, "You need to rename your mutant. BTS is the name of a Korean pop band," and I was like, I had never heard of BTS before, but but now every time I see BTS, I was like, this is why this person was upset. But yeah, no, to me, BTS stands for the Brutus mutant of Arabidopsis thaliana, is involved in iron homeostasis, and then for the Generation Z, it stands for a Korean pop band. But anyway, whatever I can do to get people interested in our work. And so I'll tell you a little bit about what we're doing currently with Popeye. I won't focus so much on Brutus in the interest of time, although I think Brutus is a really, really interesting protein. So Brutus Popeye loss of function mutants exhibit, as I said before, decreased growth, and this assay shows rhizosphere acidification. This is what's called a Bromocrestal purple assay. Basically, is the media contains a pH indicator that turns yellow when it is being acidified. And so these plants were grown under iron deficient conditions for 3 days and then transferred to this pH indicator. And as you can see, wild type plants acidify the rhizosphere, whereas Popeye mutants do not. Popeye mutants also exhibit decreased amounts of iron reductase activity, yet they contain increased amounts of iron. This suggests that plants somehow are able to bring in more iron, but they're not able to utilize that iron. And this is manifested in decreased tolerance to soil conditions in which the pH is elevated, which decreases iron bioavailability. And so we think that Popeye, normally it activates the response, and in an iron deficient, in a Popeye mutant, you can't activate that deficiency response, perhaps because the signaling mechanisms for iron within the plant are disrupted. To try to get an idea of how it might be doing that, we went back to microarray analysis, and we checked to see what sort of genes were misexpressed in a Popeye mutant. And as you can see, many of the classic responses to iron deficiency that wild-type plants usually undergo do not take place in a Popeye mutant, or they are actually enhanced in a Popeye mutant. And so on this side, you see that many genes involved in a variety of different processes are impacted in a Popeye mutant. And these indicate some of the key iron homeostasis genes that are impacted. I want to point out 3 here, ZIF, NAS, and FRO, which play a variety of different roles in changing iron availability. For example, NAS4 is involved in binding and facilitating iron transport in the phloem. So we were intrigued by these, and we performed ChIP, which is basically a way to see what cis regulatory elements or what DNA the Popeye might be binding to. And so I say this, and I forgot to mention that Popeye is actually encodes a BHLH transcription factor. So it being a BHLH transcription factor, we conducted ChIP to see what promoters Popeye might be binding to. Popeye directly targets genes involved in a number of processes: transcription, transport, photosynthesis, development, and several key iron homeostasis genes indicated in red here are actually direct Popeye targets. So as you may have mentioned, I mentioned that FIT is a BHLH transcription factor as well. And FIT, one of the master BHLH transcription factors involved in iron homeostasis, binds to its similar other BHLH proteins that are homologous. So we were intrigued by the possibility that Popeye, which is another BHLH protein, might also have binding partners. So we looked at the variety of different homologs that were closely related to Popeye. So there are approximately 133 some odd BHLH transcription factors that are encoded by the Arabidopsis genome. Many of them are related. This particular group of BHLH transcription factors are broken down into 4 different groups. And this particular group, we found that Popeye actually interacts with BHLH transcription factors that were in the subgroup here in blue. We show this using yeast to hybrid analysis and biopsy. And you can see that, for example, Popeye interacts with ILR3 and 115, specifically in the nucleus. So this biopsy shows you the Kochiana benthamiana epidermal cell, which actually is expressing an RFP tag that is nucleus specific. And biopsy allows you to see a green signal if you if two proteins interact. So you basically transiently transform the epidermal cells with a protein that's tagged with the N terminus of a fluorescent tag, and the C terminus is tagged to another protein. And if these two proteins interact, we see a green signal. And so you can see this green signal is appearing in the nucleus. This tells us that Popeye interacts with its homologs in the nucleus. And this work was done by a former postdoc in the lab, Devarshay Sagoti. And so we began to build upon the network that we started with. So we knew Popeye interacts with the promoters of these transcription factors, and I don't have time to go into it now, but it seems to be repressing these genes. We also know that Popeye is interacting with several Popeye homologs, and that it's also controlling many other genes that may not play a role in iron homeostasis. And so it interacts with and potentially represses iron uptake or represses the distribution of iron, which controls sensing, response, and bioavailability. I don't have time to go into this, but another former member of the NAB, Rosalind Samira, who's actually a postdoc at Texas Tech now, did some work on another Popeye homolog, ILR3, and actually found out it was involved in modulating glucose synthesis under iron deficiency. And so that kind of adds to the complexity of the network as well. She as well as Anna Mathiadis also spent some time looking at this other protein that I mentioned to you. It turns out that Popeye interacts with Popeye homologs. And it turns out using yeast 2 hybrid analysis and biopsy, we also found out that Brutus, the protein that shows the opposite response to iron deficiency, also interacts with these same Popeye homologs. And so each year we learn more and more about this network, and the network has grown more complex. What we became interested in as we expanded this network was, can we begin to use mathematical modeling to make predictions about the network? And so another former graduate student in my lab, Anna Mathiadis, interacted with a graduate student in the College of Engineering in Electrical and Computer Engineering, Alexander Koryachko. And Alexander was mentored by Dr. Krinos Williams, who's a fantastic collaborator here at ECE. And they use mathematical modeling and engineering concepts to try to create algorithms to understand relationships. And in this particular case, it was relationships between genes. And so this image is from a review. It talks about the different types of algorithms that one can develop to try to look at transcript data and expression data to try to figure out relationships between groups of genes. And so we started off basically with a type 3 or type 4 algorithm where you're establishing quantitative relationships between gene A and gene B, or gene 1 and gene 2, based upon expression pattern. So if I give you a time course data set, you should be able to predict what regulators are regulating things that are being expressed at a later time point. And so the algorithm that they created based upon this idea was called the cluster and differential alignment algorithm. And so this allowed us to try to predict what might be happening what might be regulating some well-known iron homeostasis genes based upon what might be expressed earlier with a similar expression pattern. So using this approach, we were able to make another layer to this regulatory network and predict several novel genes that with no known role in iron homeostasis as actually regulating some of these genes. And what I'm showing you here is the relationship between ASO2 and Brutus and Popeye and some other well-known iron homeostasis genes. So basically, to make a long story short, we said, here's the expression pattern of these genes. Can you predict what might be regulating it? And ASO2 seems to act upstream. And I'm only showing this because of the complexity of the image there. And using this information, we could then go back and create a type 5 algorithm where we could begin to describe the dynamic behavior of this network. And based upon this approach, we were able to make predictions about what would happen if you would have not single mutations, but higher order mutations between some of these genes and make predictions about how some of these higher order mutations would behave. More recently, we've begun to work specifically on Popeye from the perspective of its mobility. So this is some work done. It's been done by another former graduate student in my lab, Juri Shawar Muhammad, who's now a postdoc at Rice University with Bonnie Bartel. And she did this work with another good friend of mine, a collaborator here at NC State, Dr. Vasu Tsani. So Popeye was really interesting to us because if you look at a transcriptional fusion, you can see that Popeye is expressed predominantly in the vasculature. But if you look at a translational fusion, you find that the protein seems to be localized in all cell types. This suggests that the protein might be moving in between cells. Right? And so Jury paired up with Dr. Suzani using something called pair correlation function to try to create an understanding of how this protein might be moving in between cell types. So here's a confocal image of the Arabidopsis root. This is a really zoomed in version showing you the different cell types of vasculature, endodermis, cortex, and epidermis. And we can image the GFP signal. And in this particular case, Popeye, the protein is fused to GFP, creating this greenish signal. And we can use confocal, we can scan carefully across these images of the root and then get what's called a carpet that basically gives you a signal indicating the signal across both time and space. And working closely with another graduate student in Ross's lab, we were able to make predictions about the mobility of Popeye, indicating that it was mobile compared to another protein that we knew was not mobile. So this pair correlation intensity enabled us to say with some confidence that it's a mobile protein. And so we were really intrigued by this idea that it's a mobile protein, and we wonder what could be its function in multiple cell types. Is it doing different things in different cell types? So we created transgenic lines in which Popeye was expressed used specifically in the vasculature using a vascular specific promoter, or in the endodermis or in the epidermis. And we found in some cases that when we expressed it in certain cell types, it would move, and in some cases it would not. So in this, for example, here you see a version of a version in which it's expressed in the cortex and it moves to the endodermis, and then expressed in the cortex and moves into the endodermis and vasculature. Or it's expressed in the epidermis and it moves into the cortex. And so we were to utilize these lines as tools to make predictions, to try to probe into the function of Popeye in different cell types. And so one of the first things we did was ask, what is it doing, and what happens in terms of iron deficiency response? So here's our wall type showing rhizosphere acidification. Here's our Popeye lines with no Popeye, and here's our complemented line. We found that when Popeye is expressed in or moving only in the vasculature or in the endodermis, we don't see complementation. We also sort of see this when we look at the shoots as well. So the shoots don't grow the shoots perform very poorly in iron deficiency. And so we don't see this rhizosphere acidification, nor do we see this nice greenishness that we would like to see in our wall type shoots. However, when it's in the cortex, endodermis, and vasculature, we do see rhizosphere acidification, and we do see nice greening of the shoots. This suggests there's something about it being localized in the cortex, endodermis, and vasculature that's important for its function. But when it's expressed only in the vasculature and endodermis, then it seems to hinder the plant growth. So there's something about its localization in the cortex that's really critical. I won't go into excruciating detail here, but this is what's called ICP mass spec analysis, so that we can look at the amount of different metals in these lines. So here is iron content. The iron content is low because we're doing this experiment under low iron conditions. And under low, I should point out that low iron leads to an increase in a lot of other metals, because iron just triggers the activity of IRT1, which is a major metal iron transporter, and it acts non-specifically to uptake other metals. So quite often in iron deficiency, you'll see an increase in other metals. And what I just want to point out briefly is that in many cases, for many of the metals, we see the same amount of these other metals in a CE-NV line as we do in our Popeye mutant line. And remember, CE-NV was the line in which seems to complement physiologically the phenotype, whereas the Popeye lines do not. So it's odd that these plants perform much worse under minus iron than these plants, even though they have the same amount of other metals. So to try to get an idea of why that might be, we did Rna-Seq. Now we're trying to get a little bit more modernized. We did Rna-Seq analysis, and we found a variety of many genes were differentially expressed between the different cell types. So in our VEN line versus our CE-NV line, which is, remember, the poor growing line versus the line that's going really well, a lot of things didn't change. However, there were some things that did change. And we were really intrigued by this particular set of genes because this is one of the few sets of things that really differentiated CE-NV, which, remember, with the one that seemed to complement the phenotype, from all the other lines and was very similar to wild type. And this particular set of genes is enriched in genes involved in photosynthesis and carbon metabolism. So what we think is happening is that there's a shoot signal that's derived that goes to the vasculature. Popeye plays an important role in then communicating this signal to the other cell types. And perhaps it plays some sort of role in controlling carbon metabolism and photosynthesis in the cortex, which then facilitates its ability to respond to iron deficiency. So we think that there's something specific happening in the cortex associated with photosynthesis that Popeye seems to be controlled that's changing its tolerance to iron deficiency. And then, Ben, can you hear me? I don't know if I have much more time. I have another little vignette, but I'm not sure if I'm out of time yet. Okay. And so the last little vignette I'll tell you is about some work that my current graduate student is working on, Imani Madison. Imani was really interested in looking at iron deficiency response and how it might correlate and how it plays out in terms of in combination with heat stress. And we had these luciferase mark lines for both Brutus and Popeye that she began to play with. And so she began to ask the question, what happens when you have heat or a heat stress or a heat shock or iron deficiency or a combination of both? Normally we see an increase in Brutus expression and increase in Popeye expression under iron deficient conditions. But she was surprised to see that when you look at heat stress, you see the kind of the opposite effect, like a decrease in expression. But when you add the two together, shown in yellow, you almost see a cancellation of the impact of the two different stress conditions. So she teamed up with another talented graduate student in Kranos Williams' lab in ECE to try to dig into how that might be taking place, to what might be happening here. And so, another drink, sorry. And so what they did was they selected a marker line for the cell cycle. It's like they selected a cyclin B1 GFP marker line because they wanted to see how these different stress conditions were affecting the cell cycle and how this might play out in terms of overall growth of the plant itself. And so using this signal, they treated these plants to these different conditions. And then Eli created an automated image analysis and then a filtering pipeline to try to be able to look at this signal over time using light sheet microscopy. Right? And so this light sheet microscopy we were able to capture images over time throughout a 24-hour time point, looking at the signal for cyclin B1 in specific cell types. And so processing this image using a software that he developed called BioVision Tracker, we were able to see changes in the cell cycle responsible for heat shock, iron stress, and a combination of the two. And we did all this work in what's called the magic chamber. It's basically a small tube that allows you to grow plants like Arabidopsis plants in a clear gel, and then you can image the root using light sheet microscopy. And this magic chamber was created by Dr. Ross Sutzani, who I mentioned earlier. And so using magic in combination with light sheet microscopy and BioVision tracker, we were able to look at parameters for the cell cycle at a resolution that had never been seen before. So we were able to look at things like, the persistency of the signal over time, the appearance of the GFP signal over time, how long the signal, the ends of the signal, how long those ends persisted as the cell grew over a 24-hour period. And we were able to do this for both heat stress, iron deficiency stress, and a combination. And then integrating these signals over time, we were able to come up with a sum of square analysis that allows us to really kind of quantify how, whereas there is a response to heat, whereas there is a response to iron deficiency stress, and there is a response to heat stress, these two responses seem to cancel each other out over time, suggesting that the iron deficiency response is antagonistic to heat stress. So another final example of a war situation where you have two different stress conditions warring at war over how it controls the cell cycle. And so with that, I know it's kind of all over the place, but it's stuff that I'm interested in thinking about. And with that, I'll thank my current graduate student, Imani, and a really talented undergraduate in the lab, Chloe, the former members of the lab who've done a lot of this work, and collaborators across NC State and beyond, as well as collaborators at other institutions. I'd like to thank the National Science Foundation for funding me for a number of years, the NC Ag Research Service, and the Cal's Dean Enrichment Program for some of the work that I was doing with John McDowell. And with that, I guess I'll stop. Hopefully not everyone's asleep.
No, that was fantastic. That was really cool. I just learned a ton. Wow. That was a great talk.
I don't want to talk fast, so I apologize.
Oh, no, man, that was great. That was, yeah. I don't even have the right words. That was really cool. So first, I would like to say that there were many in the chat that when listening that laughed at the Brutus and Popeye. Yay. So you were not alone on that one, myself included. Yep, grew up watching those too, so I appreciated that.
How much time we spent arguing over what we call them Bluto or Brutus, and some of the millennial lab members were like, I don't know who Brutus is. They just only know Bluto. So anyway,
that's fair. That's fair. I remember Bluto. Yeah, but I feel like Brutus works well.
Thank you. Exactly.
All right. So we will just jump into the questions real quick here. So first, do plants? You were talking about the nutrient uptake sort of in general and how well the plant assimilates it and all that. So like, people will take multivitamins, but there's some questions about whether or not the vitamins that are in those multivitamins are actually able to be taken up. Are there similar issues for plants where, like, people try and give them a nutrient that they think they need, but it's not in the right form or something like that?
I wouldn't say that. I wouldn't be surprised if there were. I know that a lot of there's a lot of eutrophication is basically a trend that we use to describe all the nutrients that are applied that the plants don't use. There's quite a bit of nutrients that people apply that they don't use for a variety of different reasons, and that can lead to lots of environmental problems. But I wouldn't be surprised if there were not. I don't think that's a common thing. I think we pretty much worked out the types of nutrients or the ways that, for example, the different forms of nitrogen that the plant can absorb. I wouldn't be surprised, but I don't think that's very common. I don't think.
Okay, that's good. That's really fascinating. So how stable are the ferrokelatases, and are a lot of resources used in plants to regenerate the enzymes? This is one of our chatters, Arctech. He says, I know that chemists try to replicate this chemistry, but iron is very notoriously difficult to study for catalysis, and is curious how nature stacks up versus the synthetic chemists.
I don't know about the stability of ferrochelatase. It could be something that's well studied. I'm not 100% certain. It's not my area of expertise. I know that there are different mechanisms that different organelles utilize to make the iron sulfur or to
Hmm.
make the heme clusters. But I'm not sure about the stability of ferrochelatase. That's a good question.
Okay.
Yeah, yeah. I'm sorry, I don't have the answer to that one.
Oh, that's fine. So let's see. Okay, so you talked about how the plants will, like, to try and make the iron more soluble, they'll basically acidify the soil around them by releasing protons
Right.
into that soil region immediately around them. Is that, I mean, I've heard about people say, talking about, like, you need the pH of your soil to be a certain, in a certain range for different plants. Is that why, or is that
That's exactly
something
why. Yep.
additional? Okay.
Yeah, the pH really affects the availability of many nutrients. Some um so much so that it's like if I want to create a an iron deficient soil by
Hmm.
adding lime in, for example, I'm really lying when I see it. I'm making it iron deficient. I'm making it iron and deficient as well as deficient in many other things. So it's
Okay.
really not specific. The pH really affects a lot of different nutrients. The nutrient availability because of cation exchange or the ability to exchange cations with the soil particles. So that's the reason. Yes.
Okay. Interesting. Would it? I guess along the same lines, like, let's say I'm a chemical engineer, so my chemistry is not great. But let's say you could add something to the iron, like add something to the soil. Let's assume it won't hurt the plants somehow, but you could basically directly reduce that iron, the iron 3+ to 2+. Would that be useful for the plants, or is that something where, like, you could like overload the plant with too much iron?
Yeah, that's a really important. You don't want. Yeah, that's a really interesting point. I had not. I think there are people who are thinking about ways to make iron more availability, either by changing the microbes around it to change viability. I don't know. There may be people who are working on direct enzymes for iron reductases. I'm not aware of them, but there may be people.
Okay.
But you also have to be, like you said, really careful because excess iron can be really toxic.
Oh,
So
okay.
yeah, so excess iron can be toxic to the microbes in the environment as well
Mhm.
as to the plant itself because it leads to lipid peroxidation. So you want to
Okay.
really, so plants will take in as much as they need, and then in excess, they will sequester it into things like vacuoles, and it's also a big part of the cell wall.
Hmm.
And so if you have too much of it, it's just as bad as having not enough.
Okay, that makes sense. Let's see. Okay, so obviously, somebody had to ask it, so I went ahead and did it myself. Is there going to be an olive oil mutant?
I get that question a lot.
Yeah.
There is, yeah, we're working on
Okay.
it.
All
We're
right.
working.
Good. I feel like that. I feel like it's important. You
Yeah,
got
well,
Popeye, you got Brutus. You gotta get olive oil in there. Maybe Jeep. I don't know.
right. My daughter's name is Olivia,
Oh,
and my friend
nice.
Ross Susani swears to me. Then I named her Olivia because of olive oil. I was like, no, I'm not that into science. I'm not that
Makes.
much of a nerd. So
Yeah.
no.
Oh, that's funny. Oh goodness. Okay. So, Dr. Rama Kotapali wants to know. Under heat and drought conditions, some plants become resistant to viral infections, and she wants to know if you've looked at whether iron deficiency would affect this resistance.
We have not. Yeah, so that's a really interesting question because heat, because for example, certain biotropes and iron deficiency trigger similar pathways,
Hmm.
and iron deficiency can trigger um, resistance to to certain bacterial pathogens as well. I had not looked at viruses, however, I tend to if I'm doing anything in terms of pathogens, I would do something with bacteria or fungi because I'm more familiar with them. I have not looked at viruses, but I would suspect that it has something to do with the ferrochelatase and the I'm sorry, ferroptosis that I mentioned briefly in the
Hmm.
picture. so that iron availability really impacts ROS accumulation, which then too much iron makes ROS, and the ROS could kill virus. I would imagine so. Maybe that
Oh, okay, okay,
maybe that's something to do with it, and that's something that I have not looked into.
okay. That's interesting. So I guess it kind of, not really piggybacking off the question, but it kind of made me think about it. Are there instances where a plant having an iron deficiency can be a good thing? Like you showed
Yeah,
the sort of
exactly.
that warring between the heat uh, effects and the lack of iron, and it was almost like it sort of canceled itself out.
Right.
But
Yeah,
I don't know if that necessarily qualifies as good, but you know.
I would think so. I mean, iron deficiency makes it more difficult to have ROS accumulation,
Hmm.
which could be particularly problematic. Whereas an excess of iron, you may have that as a problem.
Okay.
So there might be situations where, in an effort to inhibit ROS accumulation, might want an iron deficient condition. Or, for example,
Mm.
I think I showed in that review paper how you want, oh gosh, I think this Magnaportha rice has been shown to create ferroptosis to kill the pathogens.
Hmm.
So you want a condition where you want to change the amount of RS accumulation because you want to change cell death. You want to basically change the ability to have a cell death situation for certain pathogens. So yeah, I think there are absolutely some conditions where a slight amount of deficiency might be a good thing. And that's a
Okay.
signaling thing too, as well. So iron deficiency could affect signaling processes, which then could trigger positive responses.
Oh, okay. I got you. So what? Like it could initially be a problem, but then the plant corrects itself, and so the net result is a positive.
Exactly, exactly. Because
Okay.
the plant has iron stores, and so a little bit of iron deficiency is not necessarily a bad thing as long as it has enough to recover its stores and eventually get back to
That
homeostasis.
that makes a lot of sense. Okay. Dr. Kospali wanted to follow up and ask, do you see any overexpression of WRKY transcription factors?
I have not looked at that, but I could look at if he sends me a list, I can look at them for you. If when you say overexpression, do you mean just an increase compared to an increase compared to plus iron? I could look into that. I mean, there's like thousands of genes. I mean, it depends on if you're looking at the whole root, you're looking at specific developmental zones, what
Hmm.
specific cell types you're talking about, but that's certainly something we could look into.
Okay. Might have to put you two in touch.
Yeah, if
She's
you guys,
out. She's out at the University of Oklahoma.
yeah, is it Worky 21? That might be. We've seen some interesting stuff with that one, but if it is, and so let me know.
Okay. I'll see if she follows up again. So there's a little bit of
Yeah,
that chat delay, but we'll. Dr. Koth probably likes that. She asks a lot of really good questions,
yeah,
and
fantastic. I'm glad she's having boarded
yeah.
today.
So I'll ask one more while she's maybe getting another one in. With with GMOs, is this something that could be tackled by that? Like maybe you replace, not replace the enzymes, but like could you, trying to think of the right way to ask this question? Could you use genetic modification to take like this model plant for instance and somehow improve its iron uptake or alter the iron uptake or alter the way like the condition under which the uptake happens in to help deal with issues that you might see in the wild?
Yeah, we don't talk about this a lot when we're writing papers and proposals, but it's a tricky subject because if you try many efforts to try to increase iron uptake kind of backfire because a lot of the iron uptake mechanisms are non-specific, and you end up uptaking more than
Mm,
you don't want to uptake. That's
okay.
one problem. The other problem is once you uptake the iron, that's great, but then you have to make the iron biologically available. And quite often there are what are they called, anti nutrients that will like phytates that'll chelate to iron and may make it so that's not biologically available. So you really have to think carefully about what ways that you're modifying the plant so that
Hmm.
you a don't bring in stuff that you don't want to bring in, but b make it so that when you eat it, you can actually utilize the nutrients.
Oh yeah, that's a great point. Not something I would have thought about, but that makes a lot of sense. It doesn't matter how many nutrients you have in a plant. If you eat it and can't get to it, it doesn't matter.
Exactly. Well, yeah. And the other big issue is like, for example, Brutus is really interesting because it has Brutus mutants contain more iron than wild type.
Mhm.
And so it's really interesting, but if the mutants actually are embryonic lethal. So you have so much iron that it kills it at the seedling stage, kills it at the embryonic stage.
Oh,
Oh shoot, what do we do now? What do we do?
yeah. Oh, Jesus.
So you have to be careful. It's always a balance, and it's not as simple as, oh, I'm just going to change this one gene, because
Right.
you never know
That's
what's going to
really
happen in
interesting.
mutations.
Yeah. Well, yeah. I mean, I guess it's a complex system, so
It's a complex system.
that
Yeah.
makes sense. And Dr. Kothapali said when she asking about the transcription factor, she says it was just in comparison with the iron response elements.
Yeah. I have not seen MIBs. I've mainly seen BHLHs. BHLHs seem to be the main um, group of transcription factors that are impacted, but
Mhm.
I don't see a ton of workies. Like I said, the only one that I saw that popped up was 21. The BHLHs and a couple of MIBs. But again, if you have a list of working genes that you're interested in, I could look at our data and see.
Okay. Yeah, I'll put her in touch with you just in case. That's interesting.
Yeah.
Oh, apparently, I didn't know this. She says it's recommended that you don't eat spinach with tomatoes because of the oxalates present in tomato seeds.
Exactly.
I was not aware.
Yeah, like sweet tea, the tannins and like the tannins in sweet tea can impact, very similar thing, can impact iron absorption as well.
Really?
So these anti-nutrients really impact. You can eat something and then you not really be absorbing it because of the other things that you eat.
That is wild. I've never heard either of those things.
But I'm Southern, so I can't go one day without a cup of sweet tea. So,
See, I'm like I told you, I'm born in South Carolina, and I am ashamed. I don't like sweet tea. I don't like tea. I don't know what it is. I don't know what it is. I don't like tea.
oh, that's amazing. Do you like barbecue?
Oh, love barbecue.
Okay, you've redeemed yourself.
Oh yeah. Oh, food. Look, yeah. Barbecue grits, hush puppies, collards. Yeah, all of that. Good. Yeah, don't get me wrong. Yeah. No, it's just tea. It's the tea. Don't worry.
Okay.
Yeah.
You're fine, then you're fine.
Oh yeah. I honestly, I think I'm really sensitive to bitter taste because I don't like coffee either. Like, I don't like bitter beers. Don't like tea. Don't like coffee. And I know they're not like super bitter, but it's like it's just a particular thing. I don't know.
We brew our tea pretty strong here, so it can be bitter.
Yeah. Yeah, I had to keep my southern bona fide somewhere else. That's all.
Yeah, right. I was like, oh, he's not even born in the South.
Oh man, no, everything else, biscuits, grits, all that good stuff. Yeah, like we're good. Just the sweet tea.
That's funny.
Oh goodness. All right, so I'll give everybody in the chat a couple more seconds or a little bit more time to get any final questions in. But in the meantime, I will say thank you again to Dr. Long. Like I said, this was fascinating. This is really cool.
All your guests, Ben, I'm sure. It was my pleasure.
No, I mean, look, I do say it to my guests, to all the guests that come on, but that's because they're all really cool. Like it's these talks have been fascinating because like I said, there a lot of them are outside of fields that I would or outside of my field, so I would otherwise never get to hear about it. And I'm learning a ton, and it's really cool to hear people that are excited about the work that they're doing and pushing boundaries in their own fields, and it's really neat.
Fantastic.
Yeah.
It's been my pleasure, and I really appreciate the questions.
Yeah, absolutely. Oh, we got one more real quick. Pizza Sepulcrate, which I love that name. Asked what other software have you seen being developed in the research? So I guess are there's because you showed like the BioVision tracker.
Students are working all the time to create new algorithms, new tools for trying to understand regulatory networks. Because that's the big thing. You really want to be able to try to make accurate predictions of regulatory network interactions so that you can cut the amount of candidates that you need to screen for. So I want to find out a new regulatory hub that controls iron deficiency response, for example. but the regulatory network predicted, 27 candidates. Well, shoot, which one? How now I got to spend the
Yeah.
next 2 years of my life,
Yeah.
trying to characterize these mutants. I'd like to know what's
Oh
what's
yeah.
so a lot of people are spending time trying to also process data. How can we get the most information out of
Hmm.
all this tons of RNA-seq data, DAP-seq? How can we process these data to really just get as much information that we can
Hmm.
out of them? Imaging software, we've getting more and more confocal, more and more ways of imaging systems. How can we combine different data sets, like ways to combine, the translatome data with the RNA transcriptome data, with the
Hmm.
proteomics and the metabolomics data? So there's all sorts of people who are busy, trying to figure out ways to do that. And these efforts have really been fueled, have really created broken down silos between disciplines. So I would never
Hmm.
have worked with an engineer, um, you know, 10 years ago, because I'm like, what do we have in common? What can we learn
Right.
from one another? But now it's nothing to say to contact Kranos and be like, all right, can you help me design some software to process this RNA-seq data better, or to
Right.
work with Ross to create neural networks that can process phenotyping data with genotyping data. So um, I know it's a very vague answer, but the answer is a lot.
Oh yeah. Yeah. But, I mean, I think it's a great answer because you're right. Like it seems like just even within the last decade or so, there's all like you said, like you put it really well. All the silos are starting to come down, and people are realizing that you've got to expand outside of your comfort zone and kind of to really push things ahead.
Absolutely. And engineers have been thinking about that, some of the stuff that they use. We're just now like, oh, you can do that? Oh, wow, you can model all of this with just like I'm working on a proposal now with Belinda Akpa, who's at Oak Ridge National Lab. She's
Hmm.
using mathematical modeling using what she calls tiny data. If you have small amounts, you just have a few images, or you just have, some qPCR data, and together she can put all this stuff together in a petri net modeling approach and come up with these amazing predictions that make a lot of biological sense. So now I have a
Yeah.
really specific experiment that I can do, as opposed to, I don't know, let me just try some stuff.
Oh yeah. Oh
My
yeah.
model says this, and I can do this, and so
That's really
working with
cool.
these engineers and mathematical models have been really a game changer.
Yeah, it makes a big difference. Yeah, because I've definitely been on the end where you have to shotgun the approach and just kind of say, "All right, I'm going to do 100 things," and then step back and see what worked and what didn't.
Exactly. Hoping the grant money doesn't run out while you're
Exactly.
delivering. Yeah.
That's really cool. Shoot, depending on how that all works out, maybe we can get you to come back and talk about that. That
Yeah,
sounds
that'd be
fascinating
cool.
too.
Yeah, if it gets funded, I'd love to.
Hey, sounds good,
Yeah.
man. All right, let's see if there's any more questions. Go ahead and get them in. But I think we're about done. Like I said, this was fascinating. This was really cool. Just hearing about the breadth of work that's being done and how it all kind of ties in together is fascinating. Um, As like I said, I don't really know a whole lot, but I know very very limited amounts of plant biology stuff coming into this, and
The same way I feel about chemical engineering. It's like the world to me. I'm trying though, but it's like,
hey, that's
I'm
good. Oh goodness. Well, so I'll thank everybody for coming out, and thank you again to Dr. Long for a fantastic talk. It's just really something else.
my
Um,
pleasure, man. But thank you so much for inviting me. It's been a lot of
yeah,
fun.
absolutely. And let me get you to hang around for just a minute. We'll talk just briefly,
Okay.
and
Okay.
I won't keep you up too late. I know, like I
Okay,
said.
sure.
Um, but yeah, thank you again to everybody for coming out. The talk, if you missed any of the talk will be uploaded to YouTube later on tonight. And yeah, so thank you again. We'll be back next week. We have Dr. Rachel Scholes from UC Berkeley. So we have another fantastic talk next week too, I hope. And I hope you will all come out and check it out. And in the meantime, have a good weekend, wear a mask, and be safe. Thanks, everybody.